Hi, I'm your host, Ray Dogum, and welcome to VibeCast. VibeCast is VibeBio's official podcast where we explore some of the hottest topics in drug development, investment strategies, and technology innovation. In this episode, our guest is Roy de Souza, founder and CEO of Boston-based BreakBio, which develops personalized vaccines and AI software that aids cancer treatments by identifying the difference between normal cells and cancer cells. In 1999, Roy founded an ad tech company called ZEDO, which he sold to Warner Brothers Discovery in 2021, after his late wife was diagnosed with cancer in 2017. Roy, it's a pleasure to be speaking with you today. Why don't we get started with you sharing your background and journey into cancer care and biotech?
Thank you, Ray, and thank you for having me on the podcast. It's a pleasure being here. My background has been technology. I've always loved technology and technology products. I was busy working on ZEDO when, unfortunately, in 2017, as you said, my wife was diagnosed with colon cancer. And the doctor at Memorial Sloan Kettering, Dr. Kemeny, says, in cases like this, we can't cure. We can only aim to prolong life. And that is when, I was surprised. I said, surely with all the technology we have, there must be a way to cure. And I started looking. Initially, I was looking for treatments, not looking to start a biotech company at all. But after seeing all the research, great research people do, great ideas, yet no treatments that can cure, that motivated me to pull these things together and pull out the best strategies and get to a new kind of way to cure solid cancers. So that's how I started.
And first of all, deeply sorry about your wife. I'm sure that was a difficult challenge. I know you have a couple kids as well. So I'm sure that was a primary motivation for you to get started with BreakBio.
Yeah, thank you. Yeah, very difficult to see that. Hopefully we can solve that problem.
Can you share also, since your previous company was an ad tech company, your motivation to start a biotech, how much did you have to learn throughout that process? What was sort of that like for you?
Well, I think, I had experience in starting from something and building it and building a team and motivating the team, which is a very, very important part of getting something like this done. My experience was in technology, complex, high-scale technology. We used a lot of machine learning actually to understand what consumers are doing, what the consumer behavior, what they are likely to buy. And that also proved very useful here in our platform because we use AI and machine learning to understand each patient's cancer cells. But so I had a lot of learning on the technology side. What I had to learn from scratch is cancer. What is cancer? Why does it exist? Lots of different theories, lots of different books. I just read a new one, by that guy who wrote the Obesity Code. It's called the Cancer Code. Very interesting, view on what cancer is. But I did online classes, read the textbooks, and attended conferences to come up to speed with that side. And I find it very interesting. The silver lining of seeing my wife the way she was and what happened to her just gives me this real motivation and a sense of purpose to fix the problem and solve it. And so happy to learn it, continue learning, reading, and happy to do that.
Yeah, and you mentioned there's many theories behind cancer, and it's been studied for many decades now, of course, and I guess centuries even. In some cases, there's evidence that people were aware of cancer. And the idea is a cell just continues to grow without stopping uncontrollably. Are there any theories that you've seen that maybe people have not really heard, or some kind of ideas that can trigger some interesting thoughts in people?
The one I read recently, it's not exactly a new idea, but he puts it into an interesting context. And what he says is that millions of years ago, there were single-cell organisms. And single-cell organisms don't care about anything else. Their only aim is to grow. So, they don't, for example, they don't have apoptosis. They don't, most cells in the human body are supposed to die eventually. They're not meant to stay around forever. But single cells don't have things like that. So what he says is as multicellular organisms came about, different cells specialized and got rules, and they have rules. And he says in cancer, what happens is some cells inside this multicellular organism revert to single cell life. They go back to how they were millions of years ago, and they don't care about the other cells. They just care about growing. They don't follow the rules. So he said it's he gave a very interesting example. He said there was someone in India grew a monkey's tail. Now that sounds crazy, but it would have taken 500 mutations, new genes, evolution to create a tail. How did that happen in one generation? He says the reason it happened is the DNA for the tail in the 500 genes was already there. And it was a monkey's tail because humans originally came from monkeys. And it was just one or two genes that switched off that long section of genes. And if those one or two genes go wrong, the tail comes back again. So he says in cancer, a few genes go wrong, which switch off the way the body changed, way cells change from single cells to multiple cells. And if those one or two genes that switch it off go wrong, then the cells revert to previous, point in evolution where those cells are acting like single cells. So interesting idea. Makes, different way of looking at cancer. The end result is the same. We still need to kill those cells, but, it's still an interesting thought.
It is, and a great example. Thanks for sharing that. Let's talk about BreakBio. So more specifically, I know you mentioned a little bit about it already, but what is the problem the company is working to solve?
The problem at a very simple level is that traditionally people come up with one drug to treat a cancer, right? And they treat the same drug in many, many patients, all the patients. Problem is, cancer is, it mutates and changes in some sort of evolutionary tree. So initially there might have been one problem which caused the cells to grow fast, but then one branch develops a new problem. It has the original problem and a new problem. And another branch has the original problem and a different problem. And there's multiple branches as the cancer grows. There's many, many different branches of cancer. And a drug that can kill one branch, even if it's a very large branch of the cancer, doesn't kill all the other branches because they have different problems. So what we're trying to do is target all the branches in a patient's cancer, all the clones. They call it, polyclonal. They call these different branches clones. Our aim is to target all the clones at the same time so that we can actually kill all the cancer in a patient and get real significant results. Some complete responses, lots of partial responses. And that is the problem we're trying to target. Not one drug for all patients, not one drug for each patient, not a new drug per patient, but we want 30 drugs for each patient. Because if you get 30 drugs, you will kill all the branches. Hopefully. We can't guarantee we'll kill all the branches, but the chances, the statistics of just 2 targets per patient is better. And 3 targets per patient is even better. But if you can find targets across all the different branches, kill all the branches, we should be able to cure solid cancers in at least some patients.
And getting to that point, I think, takes a lot of work and effort to understand the individual cancer, individual person's mutations. And so what are some of the challenges currently with that process, with this personalized medicine approach? And as I understand it, BreakBio is uniquely trying to develop vaccines, cancer vaccines for patients. Can you just describe some of I said a lot there, but can you describe some of the process there?
Sure. One of the challenges is, does personalized medicine as a concept work? Can you build different drugs per patient? And some people think you can, some people think you can't. We clearly think you can build different drugs per patient. And personalization comes along slowly. Today, when you get, sepsis or something, they test your blood. They analyze what kind of bacteria you have, and then they give you the antibiotics. So they're somewhat personalizing the treatment of that bacterial infection. Even in cancer, you get cancer, you do a foundation medicine report, and it lists mutations, and it suggests 1 or 2 drugs. So they're personalizing to some extent. We're going a step further. We're personalizing by finding the problems and then manufacturing drugs for those problems. So it's taking personalization. Personalization has not come from zero to where it is today, but we're improving personalization from not just identifying a few problems and seeing if there's an existing drug. We're trying to identify all the problems and then manufacture drugs for that patient. So that's the evolution. Some people, now believe in personalized medicine. There's a very large VC. We spoke to one of the biggest in the space, and they said that there are other companies doing personalized medicine, specifically Moderna and Biotech, who are doing personalized treatment. those companies tend to be more manufacturing companies. They're very good at manufacturing vaccines. So they're manufacturing vaccines for everything. It just so happens when you do a COVID vaccine, you don't need to personalize it because it's transmissible disease. The disease is the same in all patients. It jumps from patient to patient to patient. It's the same disease. They're doing RSV vaccines, again, the same in all patients. They're doing, flu vaccines, again, the same in all patients. In cancer, it's different in each patient, and in one patient, it's different across the tumors. Moderna has done a little bit of software. They don't really, because they're so good at manufacturing, they have such elegant manufacturing, that they have not really invested in what we believe is a problem, which is find the right targets. Before you personalize, you want to know what to manufacture. We don't believe the problem is so much manufacturing. The problem really is what are the targets in each patient. So we come to this in the sense of let's treat cancer, let's get real responses, which means we need to find the targets we're trying to kill. How are we going to kill these cancer cells? What targets are we going to find? So we use all kinds of different data, not just one type of data like Moderna, BioNTech use. Moderna and BioNTech come to it more like we can make vaccines for everything. So let's make some vaccines for cancer too. And they're less focused on the actual problem that we think, as I said, is finding the targets with really sophisticated software per patient. For example, we do mass spectrometry for each patient. Every single patient, we take the tumor tissue, we find the proteins, peptides actually on the outside of the cancer cells. We do mass spec to identify those peptides per patient. Huge amount of personalization and data going into our software. So that's where we differentiate.
I appreciate that. And maybe for the audience to just clarify something. So when you talk about vaccines in terms of COVID and things like that, you take those preemptively. You take them before you get sick, hopefully, so you prevents you from getting sick. With these cancer vaccines, you're actually, can you describe what that means? Do you take it before?
Yeah, Ray, that's a very good point. And, I keep forgetting that. And, vaccines normally are used to prevent, as a drug to prevent you getting something. In cancer, we inject the vaccines in the arms and the thighs. They create T cells. The T cells go and find the tumor inside. So you already have the cancer. It's a treatment for cancer. It's the word vaccine can be used for both the treatment and the prevention. I would prefer they use a different word, but the industry uses the word vaccine, so we've got to go with that. So
gotcha. Okay, thanks for clarifying that. Yeah. I appreciate that. Can you share a little bit about your company's current drug pipeline or vaccine pipeline?
Yeah, so we have been building out our software. We first downloaded some open source software that's analyzing cancer cells in 2018. We've been modifying it, improving it. We've got a great team. One thing I've understood in building a company is hire very good people. So we have a great team. And we've been building out the software, testing it, and then we applied for an IND. So we got approval from the FDA. Our IND is approved. Our first indication is colorectal cancer for patients who've started chemo but not progressed on chemo. Started first-line chemo but not progressed. So patients who are early. And the reason we've chosen colorectal is partly because that's the one I learned about first. But I learned about it because there is no good treatments. That's the cancer my wife had. No good treatments. They say we can only prolong life. We can't cure for metastatic disease. And the FDA therefore gives you much more leeway. They give you much more cooperative. They give you, because it's unmet medical need, what they call unmet medical need. They will give you a breakthrough therapy designation, fast track designation, accelerated approval much more easily. You have to prove that you're better than something that is very, very poor. So that's the indication we started with. We are writing INDs for the next solid cancers. We think that this personalized medicine will be used for all solid cancers. In the past, there was one set of drugs for lung cancer. And within lung cancer, there's one set of drugs for small cell lung cancer and non-small cell lung cancer. And different set of drugs for breast cancer and HER2 breast cancer or PR breast cancer. But here we're going to be analyzing the actual cancer cells and building the drugs, 30 of them, for each patient. So it won't be just for one cancer indication. It'll be for any patient with any solid cancer. We are focused on solid cancers for the moment. That's where we see the most need, and they are similar. So colorectal cancer first, breakthrough therapy designation by end of 2025 is our target.
For sure. I appreciate that. And solid cancers, in many ways are one of the more difficult cancers to attack. Some ways they're not. Some ways you can resect them and take them out of the body, but in many ways it's hard to get deep into the tumor tissue and kill the cancer cells themselves. Yeah. So can you explain maybe in a little bit more detail how these cancer vaccines are able to target the solid tumors so well? Or
sure, yeah. So I think T cells are a very powerful part of the immune system. The immune system has the innate immune system and the adaptive immune system. T cells are part of the adaptive immune system, and they're more powerful than antibodies. Antibodies are also part of the adaptive immune system. But T cells are cells, and they can kill other cells. And if they find a peptide on the outside of a cancer cell, one T cell is only trained to look for one target. It'll go around the body, and if it doesn't find that target, it'll do nothing for your whole life. But if it finds a target designed to kill, it binds to that target and it injects things like granzyme B into the cell and kills the cell. So T cells are a very, very clever part of the immune system. And, they're very specific. The T cell won't kill the wrong thing. It won't attack your hair and make your hair fall out if it's designed to find a protein, peptide actually. Peptide means small protein on the cancer cell. So T cells are very powerful, very targeted, and that's the core to how these cancer vaccines work. And so we have to inject things in the arm which create these T cells. And these T cells will then go and find the tumor. Your question was, how do they find the tumor? T cells are good at finding, they go around the body till they find what they're targeted at. But we do use other drugs in conjunction with T cells to disrupt the tumor microenvironment, which makes it easier for the T cells to enter the tumor. So the tumor microenvironment can sometimes prevent the immune system seeing it, getting inside. But by using existing drugs, which we know work for colorectal cancer, it helps those T cells get into the tumor. And for the next indication
that you're using, sorry to interrupt you. Those other drugs, are you referring to chemo?
Yes. So some are chemo, VEGF inhibitors, for example, and chemo. VEGF inhibitors are, very effective. They work in colorectal cancer. Bevacizumab or Avastin is one of them. Regorafenib or Stivarga is another one, approved in colorectal cancer because they have efficacy. They do things in colorectal cancer. And it so happens that we believe that they synergize with the T cells we create.
Yeah, and I've been hearing a lot about combination therapies. Moving forward, I think that's something that's a huge topic, and it's great to see that you're leveraging, the company BreakBio is leveraging this as well.
Our combination therapy is a combination therapy in the extreme. We do not just one drug or one target, we do 30 targets, so 30, almost like 30 drugs, plus existing drugs. So, the aim is to make sure you're covering all your bases and you can really kill all the different branches of cancer cells, all the different clones of branches in a patient. That's when you really get to a situation where you can get to cures. And the status of clinical trial is, it's great that the FDA approved the IND and we can inject humans. And they asked us lots of questions, very thorough. We were quite surprised about how up to speed the FDA is on software, for example, because, as we're injecting different things into different patients, they really need to understand the software of how we choose that. And they're good, the FDA. Their expertise is really quite remarkable, given that they cover so many things. And they also launched something called Project Frontrunner, which allows us to do cancer treatments early. We'll be one of the first companies, I think, using Project Frontrunner, where they say, don't try a new treatment when all treatments have failed, but try a treatment early when the patients are still strong and healthy, which is exactly what we need to do. So we're just in the process of working with the hospitals now. And fortunately, quite a few hospitals want to open this trial at their site, which is great because these are clinicians, these are doctors who are seeing patients. They understand everything that's out there, and they look at our trial and they look at what we're doing, and they say, we want that at our hospital for our patients, which is a very good indication that they think this is a good treatment and a treatment that they want to give their patients. So there was a great conference in Miami run by the Colorectal Cancer Alliance, which is an industry organization run by someone called Michael Sapienza, and fantastic conference. They had all the GI oncologists from all the top hospitals invited there, And that's where we presented the trial. And since that point, we've received a lot of interest from all the hospitals. Once the hospitals are ready, we need to, inject, get the tumor tissue, analyze, manufacture, inject the patient. So happening very soon, probably June, maybe a bit earlier. Yeah.
Okay. What does that time period look like in terms of like when you're able to, after the analysis of the patient's tumors have been completed, what is the time frame in order to manufacture and produce the resulting injection?
So when the patient signs consent, 42 days till they get their treatment. Their first injection is our target. So not too long.
That's a great target.
We would make it, we would try and make it quicker in the future. We will be able to compress manufacturing timelines with newer technologies, more automated technologies. But for the moment, 42 days from consent. So when they join the trial, 42 days. Yeah, we do get tumor tissue and all in advance. So we ask the patient to give us tumor tissue in advance. And when they're ready to join the trial, they sign consent, and then it's 42 days.
You mentioned how the FDA is getting more familiarized with software and AI and things like this. So can you describe a little bit more detail how BreakBio is leveraging AI and what sort of conversations maybe you had about the software with the FDA?
So yeah, the way we're leveraging AI and machine learning is fundamentally we need to know what's on the outside of the cancer cells. And what is on the outside of the cancer cells is determined by what's called the antigen presentation machinery. And there's multiple steps which take proteins, they chop them up into little bits and transport them onto these MHC molecules, and the MHC molecules put them on the outside of the cancer cell. And there's different steps, and those different steps work differently in different humans. So the immune system is designed so that it doesn't work the same in every human. It works differently in every human because that prevents otherwise there's a possibility that one disease could kill everyone if everyone's immune system is the same. So the immune systems are different. So the part that shows the cancer on the outside of the cell is different in each patient. And then again, as we talked about earlier, the mutations, the overexpressions in cancer cells are different as well in each patient. So both sides of that are different, which means that what's on the outside of the cancer cells in a patient is very complex and very different from each patient. So what we do is we extract everything on the outside with mass spectrometry, and we analyze it. And we use AI and machine learning to train our systems to learn what type of things are on the outside of the cancer cell. And then we use multiple data sets. We use DNA mutations to see what kind of, antigens, targets would they have created. We look at RNA overexpression. What kind of targets would they have created? And we look directly at the mass spec results to see what's created. So it's a very sophisticated system. And we use both, we use like machine learning, and we train our machine learning algorithms per patient on that patient's data. We don't look at what happened in past patients. Different. We need to know what's happening in this patient. For example, a patient may have HLA loss. They may lose one type of HLA, one HLA molecule. You get 3 HLA molecules from your mother, 3 from your father. If the cancer cells have changed, evolved, you can have loss of heterozygosity. You can have loss of one HLA molecule. Our system of AI training per patient takes that into account. Because it's lost, we don't see it on the outside, so we know we can deal with that, which is very important.
Interesting. I appreciate you sharing that. So currently, when you're looking at your company, taking a step back at a high level, and you already mentioned you have the IND, which is congratulations on that, and you're working towards clinical trials. Are there any sort of other inflection points that you're considering as a CEO and founder of BreakBio right now that you're working to navigate?
I think the biggest part of it was building the software and then getting the FDA to approve our trial. It is a complicated trial. It's early in patients. It's using sophisticated software and drugs in combination. So I think that was the biggest hurdle, really. Since that point, we find that the hospitals are very interested, which is great. And we have to finalize our last round for this trial, and we find that the VCs are interested. I think it's a good point that we have everything ready to go and we're starting, which is good. And also that personalized medicine, like Moderna has got, Moderna has got a breakthrough therapy designation in personalized medicine. Their personalization is very small, but it prevents cancer coming back. They're not using it to treat people who have cancer. They're preventing cancer coming back. But it's a proof of concept, which has excited some of the venture community because they were never quite sure whether this personalized medicine will work. And now they see Moderna doing it in the early stages. And again, as I said, Moderna is not focused on treating patients with tumors. They want to prevent, cancer coming back. But by seeing efficacy, by seeing that can work, then it's excited a lot of interest from the investors. And they now say personalized medicine is real and they want their play in the space. So I think that's another good timing for us, for sure.
What's amazing to me as well is you started this, when you started, you were using open source software, AI, to kind of get started, which is great. It's another kind of value that I'm starting to see is this idea of open science or open software and open medicine. I think that's a trend that makes a lot of sense, although there are some competing factors with the current way our corporate society works. So just to comment that, I don't know if you have any responses to that, but
Well, in the software world, years ago, you write your own code from scratch. And then, more and more, you don't write your own code. You get modules of code already written and you architect them together. And then often you take open source software and you start. So it's better always to start with something that's built, that's built and available. There's no point recreating the basic stuff. So that's why we started with open source. And, we've iterated and improved it for years and years, tested and improved it. So, our software is generations ahead of what we started with. But still, it's always better to start with something. There's no longer in software, it's no longer a good idea to start with nothing and build everything. So that's why we do that. Of course, the code we have, the software is our core IP and proprietary now, and what we do.
Sure, that makes sense. You mentioned how VCs are starting to get interested and they think there's a lot of potential here. Can you just describe again the current stage of funding for BreakBio and then how much you've raised so far?
Sure. So we've raised a few $1 million so far in our seed round, and we've done quite an amazing amount of work with that, which people really love. And now we're raising our Series A, and so we're just closing that round out in the next couple of months, and then we'll be, I think we'll be getting results, getting data by the end of 2025 with our Series A capital. So I think that'll take us all the way to data. At that point, with the breakthrough therapy designation, I think we'll have a lot of interest from big pharma, partnership opportunities, strategic opportunities. So, it may be one of the few companies that raises one round and doesn't need the second round, but we're looking at all options. We also are busy with our second indication. So we need to do the IND for the second indication, get that approved. And some, potential partners we've spoken to would like multiple indications all at once, all in parallel, which probably means a bit more capital and, they want to get to market faster.
So I know the first indication is colorectal cancer. What's the second that you're looking at?
The second indication is going to be lung cancer or breast cancer. We're just deciding between the two and working on both at the moment. So we'll see which one we submit first. Yeah. Bigger cancers. Those are,
Okay. So that makes sense. Thanks for sharing that.
Colon cancer is the second highest cause of cancer death. So in terms of the number of people dying, it's a very important cancer. But some of the other cancers affect more people. And all those people also want to have the very best and latest treatment. So I think that's why we're probably moving to one of the larger ones.
Well, I think it's a very exciting time, and we definitely look forward to more progress to see from BreakBio and other companies as well. Just wanted to ask you in general, as you're talking to VCs and other potential investors, what is your, perspective on the funding landscape right now?
We hear from people that the funding landscape is not great. Our experience has been good. We see people interested, people excited, people want to move forward. So I think it's probably changing. I think we're also in a slightly different position that we have our, our INDs approved. we're in a hot space. AI and machine learning to cure cancer is a hot space. Personalized medicine is a space that's recently been started to be proven out. So I think given that we have an IND, given that we're using AI and machine learning to get to actual, significant, successful treatments in cancer, and given that the recent proof points, we're fortunate in that our timing is good. So we see, very good interest, and the VCs have a lot of capital that they want to deploy. So I think the funding environment is good.
That's good. Great to hear. I've been hearing that a lot lately. Things are getting a lot better too.
Right.
What kind of partnerships are you seeking, if any, at the moment?
Well, we're open to a drug partnership for an anti-PD-1. There's an approved anti-PD-1 ideally, and that is one we're potentially open to. It would mean amending our IND, but that's an option. And in the longer term, we do see that our skill is in the software and in designing and this treatment per patient. Some of the big pharma companies are great with getting things out to market. You saw how Pfizer partnered with Biotech and their vaccine. It was a great partnership. They moved fast. They made hundreds of millions, maybe a billion doses in a year across the whole world. So I think we would like to partner with big pharma like that to get this scaled out across the world and across cancer indications at the appropriate time. And I think that'll be exciting.
Yeah, and I hope this conversation leads to some of those connections. I just want to thank you again, Roy, for your time. I don't have any other questions left. I just want to say I hope that the company BreakBio continues to progress, and hope that the clinical trials show some pretty great success in its data when it's revealed next year, hopefully, as you mentioned. And yeah, is there anything else you'd like to articulate to the community or share with the audience here today before we conclude?
Yeah, I think the summary for me is that it's a very exciting field with using AI and machine learning to get to cancer cures, really needed in the world. And when our software finds targets and we show the targets we find, we show it to oncologists and they say, wow, that's a very good target. They recognize the target sometimes, maybe 1 out of 30, but they are very excited to see what our software can find. And that's what gives me huge confidence that we will get real results in our data at the end of 2025.
Fantastic.
That's quite exciting.
Thank you very much again, Roy, for your time. Looking forward to talking to you soon.
Yeah. Thank you very much.